T-cell malignancies are hard to target because malignant and healthy T-cell share most surface antigen. So conventional CAR targets risk fratricide during manufacturing and profound T-cell aplasia in patients. The key to selective targeting is to exploit clonality. Mature T-cell express one of two mutually exclusive TCR beta constant region, TRBC1 or TRBC2, and the malignant T-cell clone is restricted to only one. A TRBC1-directed CAR, therefore, recognized and eliminates TRBC1 positive malignant cell, while leaving TRBC2 positive healthy T-cell compartment intact. That is the core of the selectivity. Is not a panty-targeting, it's a clonotype restricted targeting within the T-cell lineage. In our preclinical work, this translates into robust killing of TRBC1 positive target with the preservation of TRBC2 T-cell, and single cell imaging confirmed that the cytotoxicity was strictly TRBC1 dependent. The practical insight is that TRBC1 provides a therapeutic window in which meaningful anti-tumor activity can be achieved while preserving a functional T-cell compartment. The rationale for developing this therapy is based on biological T-cell receptor rearrangement. During thymic development, T-cell undergo allelic exclusion at the TCR beta locus, meaning that each mature T-cell expresses only one functional beta chain constant region, either TRBC1 or TRBC2, and this choice is irreversible and stable over time. In T-cell malignancies, because the disease is clonal, the entire tumor population uniformly retain the same TR-beta isoform. These make TRBC1 lineage specific, lineage intrinsic, and clonotype stable marker. What makes TRBC1 particularly promising is that it is not only merely overexpressed, it is structurally encoded within the TCR complex. At the same time, targeting TRBC1 preserves the TRBC2 positive healthy T-cell repertoire, thereby maintaining a proportion of adaptive immune compartment. Thus, the rationale is not selectivity, but also biological stability, reduced escape potential, and immune preservation within the same strategy. We tested the CAR T1 T-cell for fitness during manufacturing, for in vitro efficacy and specificity at single cell and also bulk culture levels, for safety against inadvertent tumor cell transductioning, and finally for in vivo anti-leukemia performance. First, we asked whether CAR T1 engineering impact the TRBC2 compartment. CAR T1 efficiently transduce beta-2 T-cell and preserve their fitness with stem cell memory-enriched phenotype and minimal exhaustion during manufacturing. We then evaluated in vitro potency and specificity using a Jurkat TLL and the H9 TCL lymphoma model. In both cases, we achieved nearly complete tumor killing by 72 hours, while TRBC1 negative SUD-HL1 cell were unaffected, demonstrating antigen specificity. We then test the effect of the expected on target of tumor toxicity on normal TRBC1 T lymphocyte in vitro. When CAR T1 T-cell were challenged against the bulk T lymphocyte, so expressing not only TRBC1 but also TRBC2 antigen, we observe an early but transient reduction in both populations, so beta-1 and beta-2 T-cell. Similarly, when we challenge CAR T1 T-cell against the TRBC1 compartment alone, we observe a reduction in both populations. But also we observe a limited persistence of CAR T1 effector cell with the CAR down modulation, suggesting limited persistence of this type of effector cell in this specific setting. A key safety question in T-cell malignancies is accidental tumor cell transduction during manufacturing. We modeled it by transduction TRBC1 positive tumor cell in the absence of effector cell and observe progress loss of CAR positive tumor cell with decrease of CAR expression with reduced viability, consistent with auto-killing rather than a clonal expansion. Importantly, CAR expression on tumor cell did not mask TRBC1 from CAR T1 effector cell recognition, suggesting that the risks associated with this event are negligible. We then conducted the single cell tracking experiment, which confirmed a rapid TRBC1 dependent killing. TRBC2 healthy T cells were completely spared, and the cytotoxicity was driven by specific antigen recognition rather than the contact duration between effector and target cell. Finally, in vivo, we tested the efficacy of CAR T1 in both minimal residual disease and high tumor burden setting. In the MRD, CAR T1 achieved rapid, durable eradication with 80% long-term overall survival, while in the high tumor burden experiment, we saw a transient but significant complete remission in early times, which result in significant disease-free survival benefit, suggesting that CAR T1 could be a bridge to transplant option for relapse refractory TLL and TCL patient. Finally, to enable a first-in-human CAR T1 trial, we successfully produced a GMP-compliant lentiviral vector batch using an inducible, scalable, closed process in collaboration with EDBAPS and the Hospital Clinic in Barcelona. The logical next step is to transfer the process to our recently approved in-house cell factory to proceed with the clinical testing of CAR T1 therapeutic strategy in patients with T-cell malignancies. However, this type of trial are very expensive, so we will need to first raise funds to cover the cost before proceeding with the first in-human trial of CAR T1.